So in the earlier video, we discussed about the chromosomal mutations, the various types, that is morphological as well as numeric. Now let us talk about gene mutations. Gene mutations can be classified into three categories. The first is known as point mutation. The second is known as inversion. And the third is frame shift mutation. Now to understand these, let us take a simple example. To understand point mutation, we can talk about sickle cell anemia. We have already discussed about sickle cell anemia when we were talking about pleiotropic genes. In sickle cell anemia, there are 1, 2, 3, 4, 5, 6, 7 and amino acid chain, and at the 6th position, glutamic acid gets replaced by valine. So normally at 6th position in normal hemoglobin chain, that is beta chain, at 6th position there is glutamic acid, it gets replaced by valine, and that results into this defect. Now what exactly happens here is the code that codes for glutamic acid is GAG. Now and we know that the codes which are coding for amino acids are read in triplets. So when it is GAG, it codes for glutamic acid. If there is a mutation at this point, that means only one base gets replaced. Then the changed base, instead of GAG, after this point mutation, that means change in only one nitrogen base. If it becomes GUG, then this was GAG was coding for glutamic acid, GUG codes for valine. So this mutation is due to just one nitrogen base changed, and that is why we are calling it point mutation. In case of inversion, the two bases or maybe just three bases of the entire code get flipped by 180. Again, if we say GAG is a code, and if these two get flipped by 180, the code will be GGA. So GAG was coding for another amino acid, and GGA obviously is going to code for another amino acid. But in both these situations, before the mutation and after the mutation, there has been no change. Again, let us talk about these two together. 1, 2, 3, 4, 5, 6, 7 and amino acids. We said 1, 2, 3, 4, 5, 6. And in case of point mutation, here GAG changed into GUG. And before this, the amino acid sequence or the amino acids which were produced were same. And after this also 7, 8 and so on, the same amino acids are synthesized. The change is only in one base pair, and that results into formation of a different amino acid. In case of inversion also, instead of GAG, we said it has changed to GGA, but the amino acid synthesized here will be something else other than the normal glutamic acid or any other amino acid. But before this and after this, there is no change. That means there is only one amino acid which is different. So in point mutation as well as in inversion, from that point, before that mutation there is no change, and after the mutation there is no change. Only wherever point or inversion takes place, only that amino acid is different from the normal one. The situation in frame shift mutation is slightly different. To understand frame shift, we will write down a sentence which will help us understand this very clearly. The statement is, why did the cat eat the rat. The reason for writing this statement is that all words have 3 alphabets. Why did the cat eat the rat? And the reason for choosing this is because our codes are also triplets. That means every triplet reads as a specific word in this statement. And if it is a genetic code, each triplet is actually coding for a specific amino acid. In frame shift mutation, which is caused due to deletion or addition of a nitrogen base. We'll take both the examples to understand what exactly happens in frame shift. Say we are talking of deletion. That means one base, nitrogen base, gets deleted. Say this D gets deleted. And now when we have to read the code, it is going to read as Y, and again we have to read it as a triplet. So the next 3 are going to be IDI, then HEC, then ATE, again ATT, HER, and so on. Now in this statement, Y means something to us, did also means something to us, but after the mutation, now the new word is IDT, which does not make any sense to us. If this happens in a code, this codes for an amino acid, DID or that code will also code for an amino acid, but after we have removed from that deleted part or deleted base, everything has shifted. The frame to read the next 3 has shifted, and that is why we call it frame shift mutation. This example is when something gets deleted. Now, if something gets added here, suppose we write one more word, X here. How are we going to read this triplet now? The first one is going to be Y, next is going to be XDI, then is DTH, ECA, and so on. So after this added base, again the frame is shifting. Whether there is a deletion or addition, there is a shift in the frame of the triplet. And that is why the name given to this is frame shift mutation. But in all 3, the mutation takes place at one nitrogen base, and that is why it is known as a gene mutation. These are the 3 types. So this is the main method by which we classify the mutations, that is chromosomal and gene mutation. Now if we want to classify mutations in some other ways, then we can classify it on the basis of the type of cells in which this mutation is taking place. So the next type of classification could be somatic mutations. Somatic mutations means the mutations which take place in the somatic or vegetative cells. Whereas the second category would be germinal. Germinal cells or the germinal epithelium is found in the lining of gonads. And these are the cells which are responsible for formation of the gametes. That means if the germinal cells undergo mutations, then these mutations get inherited from the parent generation to the offsprings, because these are the cells which are responsible for formation of the gametes. Whereas if somatic mutation takes place, that means if somebody gets skin cancer due to mutation, then that skin cancer is not passed on to the offspring, because the mutation that took place was in the vegetative cell or somatic cell. So this is another way of classifying mutations. And we have to remember that mutations are invariably harmful mutations. So to classify, we can take chromosomal or gene mutation, or the cells in which these mutations can take place. Another way of classifying these mutations could be spontaneous Spontaneous mutation is the normal mutation which takes place in nature on its own, and it is always spontaneous or sudden. And the second category could be induced mutation. And these induced mutations are the ones which we induce in plants using various mutagens. The mutagens which we discussed in the previous video, all the chemicals or radiations which can cause mutation. So it can be spontaneous, it can happen on its own because of, again, it happens due to some mutagens only, but without our intervention into it, or we purposefully doing it. Here we are introducing that mutagen which causes or induces that particular mutation. So this is another way of classifying mutation. Let us talk about one more type of mutation. That is one more way by which we can classify these mutations. And this is on the basis of what is the outcome of that mutation. So we can classify it as same sense mutation, then anti-sense mutation, and then no sense or non-sense mutation. Now here let us take an example. The example is, say, GAG. We said GAG is a code which codes for glutamic acid. Glutamic acid. If this mutation takes place in this code, and after mutation the code reads as GCG, that means at this position there is a change. Instead of A, there is C now. This code also codes for glutamic acid. So, though there is a mutation, but it means the same amino acid. So, though there is a mutation, but it is not going to affect in any ways, because originally also it was responsible for formation of glutamic acid, and after this mutation also the acid or amino acid which is formed is glutamic acid, and that is why we call it same sense. Again, in case of anti-sense, let us take the same example, GAG coding for glutamic acid, and when this undergoes, again we are talking of the same position, that is the middle position here. If the mutation takes place and it becomes GUG, we just now talked about this, this codes for valine. So here there is a different amino acid which gets synthesized. So this is known as anti-sense. That means it makes a different sense altogether. And non-sense mutation means, say if we are talking of GAG again, it codes for glutamic acid. Instead, if there is a non-sense codon which comes in its position, that means suppose if we take example UAG, here the mutation is taking place at the first position. So if it is UAG, it is a non-sense codon. That means if it was GAG, it was coding for glutamic acid, and now because of mutation it has become UAG, which actually means that there is no code. It is a nonsense codon or a stop codon. That means there is no amino acid which is going to come at this position. So this is one more way of classifying our mutations. So we have seen all various types of different mutations and the ways in which we classify them.